Welcome everybody, and thank you so much for attending this external UCLTB seminar. As you know, we try and run these about twice a year, and we sit down in advance to think about who we would like to speak. And we wanted Di to speak for ages. She didn't know that, of course. When we asked her, she was kind enough to say yes straight away. But I've admired Di as a clinician and as a researcher for years and years. I won't say how many years, but a long time. And she's phenomenal to my mind, both because she sees what are key issues in pediatric clinical care, and then through her incredible work across a variety of infectious diseases right across the globe now, has taken key questions and answered them with really, really hardcore evidence. As you'll all know, that's been recognized both through chairs here at UCL and also becoming a fellow of the Academy of Medical Sciences last year, which is such a great honor and something that we at UCL are so proud of. Some of the work that we wanted Di to talk about was the exciting idea about how do you run clinical trials in children, always not easy when it's in things like tuberculosis, during another pandemic, the new one of COVID-19. And of course, I'm trying to distinguish that we've already had pandemics before, both HIV and TB, areas where Di is particularly skilled. And what Di is going to do over the next half an hour or so is discuss how to run trials during this time, and also present data that really has shaped the future global healthcare policy for children with or at risk of tuberculosis. One thing for the panelists, welcome of course, and if you could put your questions into the Q&A box, I will then keep an eye on them and then field them for Di at the end of her presentation. So I'll stop talking now. This is being recorded, by the way, so we hope to then post it on our website later. Di, welcome, and thank you so much for giving us your time and your presentation.
Oh, thank you very much, Mark, and really a real privilege and a pleasure to be here, and thank you for your very kind words. So I entitled my talk Paediatric TB Trials During a Pandemic, and I hope what I'm going to do is really take you through some of the things that have happened in the last 18 months with the trials that we're doing at the MRC Trials Unit in Paediatric TB. So by way of an outline, I'm going to say a few words about how COVID affects children, the indirect effects on children, focusing on TB, and really just focus on telling you the stories around the trials that we've been doing, saying a bit more about the SHINE trial, which was finishing as the pandemic hit, TB CHAMP, which is recruiting and ongoing, and the SHORE trial in TB meningitis in children, which is trying to start. So first of all, saying a few words about COVID in children. I think we all know that children get COVID, that they can transmit it, but their serious illness is really very rare. 1 in 100,000 admitted to hospital, deaths even rarer. Reasons why that aren't completely clear, but probably related to children's immune systems. The pediatric inflammatory multisystem syndrome, PIMS, also quite rare, first described really in the UK, occurring after COVID in children, and now that's been validated across the world. But again, most kids do pretty well with supportive care. And long COVID can occur in teenagers and is a worry, but it's probably less common than it is in adults. But what about the indirect effects of COVID in children? And really, I mean, we saw some of that in reports after Ebola about how children were affected by pandemics. And this could apply to any pandemic, probably because of things like the impacts on maternal child health services, including pregnancy care, immunizations, and routine child health services. And also because kids, when they get sick, they get acutely sick. with things like meningitis and pneumonia need rapid treatment. And so that suffers, and you get late presentations of things like TB and HIV for all the reasons around lockdowns in particular, and effects of lockdowns on transport, curfews, and fears of coming to hospitals. The economic effects will be most badly hit for children who are very vulnerable to poverty and malnutrition. We've heard in all countries about school closures, and of course, they're going to be the last to be vaccinated. And we just have to think about that a bit. I mean, a lot of the countries we work in where vaccination rates against COVID are very low, but then countries like Malawi, half the population are children, and where there's still lots of discussions about benefits of COVID vaccinations in children as individuals versus public health. A recent report that's just come out showing 1.5 million children out of school during the pandemic, with a third with no access to remote learning, and school systems really being at risk of collapse, especially in Africa. And these are safe havens for children. Girls particularly can be affected. And we know, have had discussions about the effects on mental health of teenagers not being able to see their peers, and also worrying about perhaps transmitting COVID in multi-generational households. So I'm going to say a little bit more about TB. Here we are, the global TB report, which came out in 2020. That is mainly referring to what's happened before the pandemic, so during 2019. But here is a picture of what happened in India after their first lockdown. And this is 14 high burden countries across the world to TB reporting. And they included in that report some data on the impacts of different countries, including high burden countries, on health service availability for people with TB, reallocation of TB resources because of COVID. So that's both staff, funding, and then gene expert in machines in the case of TB. And some of the mitigations, of course, can have benefits. I mean, including perhaps this talks, which can be done remotely in terms of training. And we've quickly learned to use Zoom and digital technologies. But there is the digital divide. But on the ground, providing multi-month medicines and home delivery and enabling others to collect drugs have been important components, which we've also adopted within the trials. Now, many of you know that the UN General Assembly had a high-level meeting in 2018, had a roadmap for TB in children and adolescents who've been left behind, but still, and they also committed to trying to treat 3.5 million children with TB by 2022, and that means, of course, diagnosing them. But in that 2020 report, 12% of cases are in children, only 8% in 2019, so this was pre-pandemic, of notifications involved children. 16% of deaths, nearly all in untreated children, because children do very well if they are treated. And there's a large TB prevention gap, particularly in under 5 children who are exposed to adult contacts. And BCG coverage remains rather precarious. In 2020, in October, the Union warned that COVID pandemic mustn't divert from the needs of children and adolescents in TB and endemic African countries, because the TB, which really is going to affect far more children than COVID, has only recently been afforded the attention which was long overdue. And certainly BCG supplies have been disrupted. When COVID was a big problem in India, then manufacture was affected, and coverage has been shown to be decreased in Africa and Pakistan. And one modeling exercise by Richard White recently presented at the virtual Paediatric TB meeting last week suggests that up to 17% increase in childhood deaths at the beginning of the epidemic. That was with the first wave. So I'm now going to move on to say something about our TB trials. So they're part of the respiratory infections program at the MRC CTU at UCL. And these are the 3 trials I'm going to talk about: SHINE, TB Champ, and SURE. And they're at different stages when the pandemic hit. They're part of the respiratory infections program. Predominantly, what you see in the red box are the TB trials, which are quite a lot of adult trials, but increasingly, especially when we're talking about latent TB or early treatment, we're thinking about doing adult and children trials together. And that's something, as a pediatrician, I really welcome. Why have children generally been left out of TB trials? Because there are not many phase 3 trials worldwide in children. Well, we know they're less infectious. They will have paucibacillary disease, so it's seen as less of a priority from a public health point of view. Difficulties with confirming the diagnosis and measuring endpoints and getting children to cough up sputum and then try to find it within that sputum. And in general, there's been effective therapy for drug-susceptible TB. But on the other hand, the natural history does differ from adults. We could be over-treating quite a lot of mild childhood TB. And I believe children have the right to benefit from child-focused research, which gives them the best treatments possible, as much as adults. This was a nice picture from James Seddon's paper in 2018, showing that in young children, you both get more disseminated disease, but also more lymph node disease. And then as you go through adolescence, you get more adult type disease where you start getting cavities. But 2/3 of children have minimal TB or non-severe TB, which is defined and has been defined by Wiseman and Mary as a respiratory disease involving less than or equal to 1 lobe, no cavities, no airways obstruction, no miliary TB, and peripheral lymph node TB is also a feature, and they may have both. So the first trial, which was finishing when COVID started, is the SHINE trial, and this is the first large phase 3 trial of non-inferiority trial comparing 6 months treatment to 4 months treatment in children with smear-negative non-severe TB. And this was just showing you the schema here. So we had 600 children randomized to 6 months and 600 to 4 months. And what we had decreased was the continuation phase of TB treatment, follow up for 72 weeks, and within which we nested PK studies of, at that time, because this started back in 2016, new dispersible fixed dose formulations, which were dispensed according to simple WHO weight bands. These were the countries, so Cape Town, Lusaka, Kampala in Africa, and then 2 centers in Chennai. And we also worked on the PK work with colleagues in the Netherlands. Children under 16 with non-severe TB who were smear negative, gene expert positivity was allowed. They had to have known HIV, but they could have HIV infection. So we had broad inclusion criteria and a decision to treat with first-line treatment and no evidence of resistance. And our primary endpoint was TB treatment failure, relapse or reinfection or death at 72 weeks, and we had a number of secondary endpoints. And just to show that, in fact, this wonderful group of sites were extremely good at enrolling. We put the first child in July 2016, planned for 2 years recruitment, and you almost can't see the red line on top of the blue one. So recruitment was done really on time, so we could enroll in trials on time without a pandemic. And the last child actually completed follow-up in January 2020, so just before the wave started to hit us and Africa. So you could say we had plenty of time during lockdown to put the data together, lock the database, which was locked in about 4.5 months later. And then in mid-June, we were looking forward to having our results meeting, which we hoped was going to be in, I think it was going to be in Uganda, actually, because we rotate going to the different sites for these annual meetings usually. And it had to be virtual. So, of course, we'd all learnt just about how to use Zoom by then. And we had our virtual meeting in June 2020. We had to remind ourselves of the good times. So here we were when we all met together previously in the trial management group in Lusaka. Here we were at the beginning of the kickoff in Cape Town, London with all our coats on. When the IULT was in Liverpool, we had a meeting just beforehand. Lovely visits to India with ongoing Penta training alongside meetings. And then going to Kampala, that was another trial management group, and all going out to a very nice dinner. And we met some of the children there, and there's Anna took over with the kids. And we were very much looking forward to the matching colours of Shine, matching the Seville oranges, when we realised we weren't going to be able to present in person either. So we had to join the virtual IUALT 2020 meeting, and Eric from Uganda presented the results of the Shine trial there. But just to give you a few slides on those results, we had very good retention and adherence to randomized duration throughout the trial. And a word about analysis. We did 3 analyses. This was a non-inferiority trial. We have modified intention to treat, where we actually excluded late screening failures and those who didn't reach week 16, because that was the intensive phase and all children were treated the same during that first 16 weeks. We had a per protocol analysis and an intention to treat analysis. And one of the things that we did, because of this issue about how do you diagnose TB in children, was we had a key secondary analysis where we had an independent expert committee, wonderful folk who helped us to adjudicate, blind to randomized arm, which children they thought really had got TB. And that was assumed to be 80% of children. So we overpowered the trial. And we had a 6% non-inferiority margin, assumed 10% lost follow-up, which we didn't have nearly that amount, and 90% power. So these were some of the baseline characteristics. Children about 3.5, 10% had HIV, and 14% had lab evidence of TB, and they had a mixture of respiratory and lymph node TB as expected. And our outcomes, just to orientate yourself, the vertical line here shows you the 6% non-inferiority margin. Here is 0 here, and you can see that whichever kind of analysis we did, we saw non-inferiority nicely shown, including in those adjudicated to have TB at baseline. And improved specified subgroup analyses similarly show consistent findings. And we did an economic analysis alongside the trial, not because we thought that 6 months treatment will be more expensive than 4 months, but to actually try and estimate the kind of healthcare costs you'd have per child. This was within trial, this economic analysis is part of the main paper. But there will be further economic modelling because implementation issues are very important. So in summary, we found that 4 months was as good as 6 months for smear-negative non-severe TB in children, with few unfavourable outcomes, similar in both arms, and reduced healthcare costs. How has COVID affected Chime? Well, not that much. Everything has been virtual, including the results meeting, so we haven't had our party yet. We've had delays in some of the PK work, particularly in India, which was hit by a bad wave of COVID, and in being able to sort out quality control with our Netherlands labs and between Cape Town and India, and moving samples. And also some of the issues around analyses and sorting the data, because clinicians were diverted to doing working on COVID. There's work in Bristol looking at a latent class analysis to try and identify those children with minimal TB using our baseline data, and with York, because they were tied up with COVID work. And also we've had remote working, pretty hard for statisticians, especially living in small flats and trying to do it on their laptops on their beds. Just showing you that timeline again. We had quite a lot of other sub-study presentations at the Union. Main results have been submitted or under review. Hopefully we'll hear soon, and the African PK results have been accepted for publication. And we also, June 2021, presented results to the WHO GDG, who held their meeting a little bit later than usual, and we were able to present those results there. And in fact, they put out a rapid communication just last month for children under 16 with non-severe presumed drug-sensitive TB, that a 4-month regimen should be used rather than a 6-month regimen. Their full guidelines will be out at the end of the year, along with an operational handbook which focuses on some of the implementation considerations. But just a couple of things about implementation, which is always important, always dear to our hearts, because why would we do the trials otherwise? If smears are being replaced by expert, we did look and find that children who were expert positive in the trial had either low or very low positivity. And these could be characterized as having minimal TB. We also have a big question about X-rays. I mean, minimal TB was defined by having little chest X-ray involvement. And what about if chest X-rays are not available? So some work going on in that is a chest X-ray toolkit and infographics, exploring the role of digital automated chest X-ray reading with Hanif, in fact, and with FIND, and further economics work to look at what the savings might be, which you could use to diagnose minimal TB, as well as I mentioned before, the latent class analysis, whether there are some clinical things in our very rich data set that may be helpful to look at to help with this implementation issue. So I'm going to move on now to our next trial, which is TB Champ, which is a prevention trial. The lead of that trial is Annika Hessling. It's been done in several sites in South Africa, and it was ongoing during COVID-19. And just as by way of a little bit of background, there's quite a lot of exposure for children to MDR-TB, and a number will progress to TB disease. It's unclear what may be beneficial, and at the time when this trial was started, WHO stated the need for quality evidence. And TB Champ is one of 3 phase 3 trials to evaluate this. VQuin is nearly all adults, being done in Vietnam, comparing levofloxacin with placebo. Phoenix has recently started comparing levofloxacin with isoniazid in adults and children, and TB Champ, where is mainly under 5-year-olds, is levo versus placebo. So this is the question: is levo for 6 months effective in preventing MDR-TB in high-risk children from MDR household contact? And it's a phase 3 cluster randomized placebo-controlled trial. Well, we aim to include around 1,000 children from as many households as needed. We estimated at the time maybe half that number of households. randomize them 1-to-1 to 24 weeks of levo versus placebo, and follow for primary outcome of TB disease at 48 weeks. And these are the three sites that have been set up and running. We've got 2 more that are recently set up, quite challenging during the pandemic, where everyone's working remotely, and are about to start enrolling probably in the next week or so. It's a complicated trial. There's multiple stages, and the teams have been amazing. They've got to access data from the adults, visit the families at home, pre-screen and screen the children, explain about the trial, gain consent, randomize, get the drug to the family, follow the child, and get information if the child gets sick. So this is a little graph of what's happened with recruitment. And in fact, it was already a challenging trial to enroll to. And one of the clinicians at Annika's Centre has written a paper about this. And you can see where I've said funding there. There was actually a gap where further funding was obtained from another source during the trial, and we had a bit of a gap in enrolment. And then COVID hit, and there was another gap where recruitment was temporarily stopped, as it was in so many countries. Trials were stopped recruitment rather abruptly when COVID waves hit. And then you could see another COVID wave a bit further on. But the status is now that 3/4 of the children are recruited. Final analysis should be mid-2022. There were fewer child contacts under 5 years in the households than anticipated, and we are expanding to older children. And there are also some levels of possible rifampicin monoresistance, which were a bit higher than expected. I haven't gone into all the issues around the recruitment for this challenging trial, but COVID-19 was certainly a difficult issue. And in the words of Annika, there was less access to TB care testing or TPT. They had to put in special applications to keep the trials going early in the pandemic, arguing that children at least got care that way. They had to get permissions for traveling. There was escalating poverty. Community violence was very difficult because community-based trials where the teams were going out on the ground, that was not safe in a number of situations. So there were quite a lot of downsides, but on the plus side, she said that trials were the main source of care, motivating for both families and staff. Provision of PPE and also vaccines for the staff and PPE for families. They learnt to do remote consultations and multi-month treatment and efficient virtual meetings and training. The other thing that's happened is that we've got a lower TB incidence than anticipated. And we've worked with the VQuIN group to plan a joint analysis using a Bayesian approach. And we've done this. This is a fairly new methodology developed at the unit. We've done it with an HIV trial where we had a small group of very young children with HIV and a much larger trial with older children, where we borrowed information from the older children. And I think this is going to be a way to go in the future. This just shows TB Champ versus VQuIN. They enrolled nearly over 2,000 patients and have nearly, they will finish their trial next year. So it's a larger trial. And what we plan to do is to combine the two using a Bayesian approach, which really is about borrowing information, but taking account of the fact that there may be differences in the treatment effects because in the two different populations, and you try to set some priors of what those differences might be. And Becky Turner here has a paper on this, which has just been accepted for BMC research. So that's the way forward, we think, with TB Champ. And that was our last meeting just before the pandemic. And it was very sad, actually, because we really were motivated up to everyone was motivated to get really moving with recruitment, had all sorts of strategies, and then COVID happened. And my final trial I'm going to talk about is the SHORE trial, which is short intensified treatment for children with tuberculosis meningitis. Idea actually originally from James Seddon to do this trial, and it's a randomized trial of 6 months intensified anti-TB treatment and 2 months anti-inflammatory treatment for both HIV infected and uninfected African and Asian children. And it was near to starting when COVID hit. It's funded by the Joint Global Trials Group. So the background, very briefly, is that you have a 12-month, not very optimal regimen recommended by WHO for TB meningitis in children. And there's emerging evidence that higher doses of rifampicin would be better, using an intensified treatment with 4 drugs, and there's a place perhaps for fluoroquinolones in here. So we're using levofloxacin. And the questions were whether we could treat for 6 months rather than 12, and improve developmental outcomes in children with using anti-inflammatory doses of aspirin, following the data of Guy Thwaites from Vietnam. So here is the schema of the trial. Open label for the 6-month intensive doses and placebo controlled for the aspirin. So quite a lot of setting up of this trial had happened before the COVID pandemic hit. These were the sites planned to start in 3 sites in Africa to start with, and maybe further satellites. They have lower rates of TB meningitis. Added on Indian sites actually after we got the original grant, Chandigarh and Delhi, where they have quite high numbers of children with TB meningitis. And we were working with colleagues in Hanoi and Ho Chi Minh City at the Vietnam Wellcome Trust Unit. So what has happened? Well, first of all, we were delayed with opening the trial. I'll say a bit more about that. We eventually opened at the beginning of this year in 5 sites across Africa and Vietnam. The Indian sites have not yet opened, but almost as soon as they'd opened, there were waves across Vietnam and Africa, and recruitment was stopped by regulatory authorities in Zambia and Vietnam. In fact, in Vietnam, the children's wards were taken over by COVID. So we've only had a total of 14 months of recruitment in these 5 sites. And as you can see down here, we just started slowly going, and we're completely flat at the moment. We've looked at some of what was happening with screening, and there are other issues as well. I mean, for example, in Vietnam, it was really quite difficult to recruit children before they'd already been started on treatment. They also were having older and bigger children than we expected. We expected this mainly to be in young children. Not surprising, a lot of bacterial meningitis and other diagnoses rather than TB meningitis. But refusals for lumbar punctures is quite common particularly we saw it in Zimbabwe and Zambia. So we've got a protocol amendment going through which allows an increase in age and weight and longer time on ATT, and educational materials and a film for families about doing lumbar punctures. Where are we now? We were supposed to have opened actually in June 2020, and we didn't start opening until earlier this year. We had waves of COVID in the UK, then the African countries, then India, then Vietnam, then India, then Africa again. Delays in signing contracts at sites because of COVID and recruiting staff at sites when everyone's working remotely. Halting of any recruitment to trials by regulatory and ethics committees, particularly where trials haven't started. Diversion of staff due to COVID, and then of course, families not coming to hospitals with their sick children. And at CTU, we've had delays with slower contracts and insurance where contracts departments have been focused on COVID, and diversion of our staff to work on COVID trials as well. We may want to do something about keeping an eye on the adult TBM trials, which are just starting, like TB Intense. One thing I've noticed is that they're all using slightly different weight bands, doses, etc., and how I think it will be really important to start a conversation across these different trials in order to form policy in a coherent way. And here is the SURE team. We met at the International Union in India in October 2019 in preparation for starting in 2020, which we never did. But I remain positive and hopeful. Funding will definitely be an issue. I think it's an issue with many trials. And so I think hopefully I've demonstrated the need for TB trials to focus on gaps in knowledge. We need innovation in terms of borrowing information and working closely with adults. A lot of the impact of the COVID pandemic on children has been indirect but substantial. And I think the impact on our research has varied by where the trial is when the waves have come, and where the waves of COVID are when moving around the world. I just want to finish with what will COVID-19's lasting impact be on clinical trials? Because I've described a rather negative story, but on the other hand, we should get better at being very nimble at setting up trials fast. We've done it with COVID. Adaptive trials is now being adopted really much more widely. Why do we take, are we able to be more nimble about setting up trials? I think I've got question marks still sitting there. So I'd like to thank all the teams at CTU, and clinical teams at all the sites. They've been wonderful, and a few people who've lent me their slides, which I've adapted appropriately, hopefully. Particularly acknowledgements for the SHINE teams here, who are near the end of our journey, hopefully, and but I'm sure there'll be more work coming out of the SHINE data. And any questions? Thank you very much.
Di, thank you so much indeed. Absolutely fantastic. And as you say, really very interesting. Do you want to stop sharing your screen if that's possible? Yes. Thank you. That's great. I'll read out just while you're collecting your thoughts. There was a relatively simple question, I think, which came from Willem, who just wanted a little bit of clarification. If you could expand on the issue of expert versus smear within SHINE, and just how what you were saying about that, please.
Right. Sorry, I probably went across it rather fast. So basically, children were all smear negative, and what we were trying to do is look back, but some of them were expert positive, and we looked back and said, well, if they were expert positive, then you could look at that in a quantitative way. How much were they positive? And it was all low or very low. So we were just trying to actually use that to say, well, you shouldn't say that you cannot put a child onto 4 months if they have all the of minimal or less symptomatic TB, but they are expert positive if they are low expert positive. I don't know, Anna Takova, I think, is in the audience. She might want to say something more about that.
So let me just see. Anna, are you there? Do you want to say something? And if so, can you unmute yourself?
We actually did a sub-study on looking at expert ultra as well in Uganda, where you did see quite high sensitivity of that. And so I guess that will be something else to think about if expert ultra really comes in a big way.
I'm just gonna, there's a question from Junaid Subrati, and I'll read it to you. It says, I did a project on the effectiveness of the TB trial for pediatric children in the Southeast Asia region. However, how more effective would the SHINE trial be for children with TB in the same region? So what we're saying there is that using a very modern drug combination within TB, and comparing that to a shorter course of more standard therapy. Is that an easy question to answer, or one that would take some time? What do you think, Guy?
Well, I can say something off the top of my head. I mean, I think what we're saying is that for the majority of children, you know, sort of 2/3 of the children, especially if you're picking them up earlier than one might do. And I think that is a key thing, that, you know, they respond well, and they respond well to the standard therapies, and you only need to give it for 4 months. I mean, we're thinking about dissemination materials when this trial's published, and one thing I know is that you know, some of the mothers have said that actually 6 months is quite a long time. It would make quite a difference to be able to stop a bit earlier than that. So I guess what I'm understanding is that if you're talking about these modern drugs, that you may not need them for drug-sensitive TB in children in order to be able to reduce down to 4 months. And maybe we could reduce even further. And I think a lot of it is about picking up TB earlier in children. And one of the things that happened at the sites and at our visit to Uganda, I remember very well, they worked hand in hand with Ministry of Health. The union was trying to put in place, or help put in place, better diagnosis of TB at peripheral health centres. And it was a lack of understanding that, you know, you need to be picking up TB earlier, and that when it's picked up earlier, children aren't so sick. And people on the ground just said, well, they can't have TB. They haven't got all awful things. They aren't nearly dead, you know. Of course, that is I think, a very key thing. And we did, you know, hear of stories of children who'd had symptoms for a very long time. And by that time, they can be quite sick. If you pick it up earlier diagnosis is an absolute key. And it's really the right time to be talking about that. Along with earlier diagnosis in adults, because of course not all adults are necessarily got 4 pluses of smear positive, and this more approach of looking at subgroups in adults as well may be relevant, where you look at those who've got less smear positivity, and we know from your meta-analysis that they could be treated for a shorter period of time. So I think that's where things are moving, and hopefully in adults and children as well. The new drugs sound great. Maybe we can get away with 1 month's treatment.
Yeah, even shorter.
You know, and I think that's the thing that will need to happen in future, is trials which both look at new regimens that might be better and shorter duration across the board for adults and children. Sorry, that was a long explanation.
No, that and just following on from that, do you, in the population where you had the clinical adjudicators reviewing children based on clinical symptoms, but not necessarily positive cultures or microbiology, what was, I missed it, the outcome was similar, or was it worse?
Yes, so it was, I mean, so basically, the clinical adjudicators, and I'm sorry I skipped over that a little bit fast as well. Basically, we knew, I mean, you know, one of the criticisms has always been, well, how do you know these kids have got TB? And so we said, well, let's over-enroll and let's power the trial, assuming that 20% haven't got TB, and we'll do a key analysis on the 80% that we think have got TB. That's what we guessed anyway. So very kindly, James Seddon, we had 3 adjudicators who were kind of expert in understanding and looking at diagnosis of TB. And they looked at all the data, including any lab data, everything they could, basically, the clinical data, everything else. And we let them look at some of the response to treatment as well. And we did that both with and without. And they, it came up with a number of around 80% of the total population they felt had got TB. And the non-inferiority was exactly the same.
Okay.
Subpopulation.
Thank you. So 2 questions from Oscar de la Pascua, which start to probe into some of the pharmacology and dosing in children. One is pointing out that in children at the moment, it's relying on an empirical dose selection, showing that the current WHO weight bands do not warrant optimal exposure. What might be needed to change the concept of weight bands and selection of partner drugs to progress into confirmatory trials? In other words, trying to be simple, is it too simple? And if you feel that is the case, how might you be able to influence it?
Okay, so I mean, so just to go back in the story of children, I think it was 2010, based on a sort of meta-analysis and going right the way through the literature, it was felt that the doses were too low for children based on the PK data that was out there. And that was when WHO suggested that the doses should be higher in kids. And they had a group look at what kind of weight bands would be appropriate. Now, I remember this because we did the same thing with HIV, and I was dearly hoping that the HIV and the WHO weight bands would overlap 1% for young children, because that would make life a lot easier. But unfortunately, they came up with slightly different weight bands, and it is difficult to fit things exactly. So, in fact, the fixed dose combination dispersible tablets, which are very easy for kids to take and which we used for the first time in this trial and have since been rolled out. We've done a PK sub-study, Oscar, and it's just been published. I don't know whether it's completely out in the public domain, but it's certainly been accepted by CID. And we did that in quite a large number of children in both Africa and India. And it's true that the rifampicin levels in the youngest children are still quite low. So here you've got an issue. Well, they seem to do well. So what do you need to increase that rifampicin level so that it's in line with the rifampicin that you'd like to have and that you've got in terms of the PK levels in the older children? But I mean, I think it's again a bit of a moving target. The rifampicin doses keep going up in the adults as well. And certainly, you know, if you want to get rifampicin into CSF, we're going up to really quite high levels, as are the adult trials. So I think I'm a bit of a pragmatist, really. And, you know, you need weight bands and simple dosing, or the kids won't take it. And we know we've got good data to show that half the kids don't finish their courses when they're 6 months long and when they're complicated. And we know from our colleagues in the north that different liquids of different bottles, etc., is a nightmare for kids. And the way forward is fixed dose combinations of dissolvable tablets given according to weight bands. And I think that's a worldwide thing, actually, and we're beginning to get there with, and hopefully other diseases will follow.
Thank you. And just to say, you've had lots of comments saying what a great talk, and Anna typed to say that they haven't got a microphone. So thank you very much, Di, for answering the question that you'd raised. That brings us on to the treatment shortening. There's TBTC31 was the adult study, which looked at 4 months versus 6 months. And I still wonder if there will be actually an increased rate of relapse or frequency of relapse in the shorter course. It just, when I look at the curves, I find they appear to diverge after a period of time. And of course, that's one of the concerns, I guess, with the short course regimen also in children. And then thinking about TB meningitis, which there's been a few questions on, the treatment duration of 6 months, you must have initially probably thought to yourself, goodness, where's this going to take us compared to 12? And there's a question again from Oscar saying, how do you decide what sort of preclinical studies do you think are needed for us to get to a sort of optimal dosing, both dose, which you've described, and also duration? Are there ways that we could do better than just say, oh, I think that'll probably work?
Yeah, well, that's a few different questions, but I think just taking the story of TB meningitis, I guess, again, you've got a story of wanting to treat kids earlier. And it's not just about the anti-TB drugs either. So Kelly Dooley from the US has been doing a TB meningitis trial and has actually done some PK work within that, looking at higher doses of rifampicin as well. So we already have some ideas about, those higher doses. Certainly could do with some more data. And for TB, it's really quite important to try and get CSF levels as well. Actually, and certainly in Chandigarh, for example, they're in a pediatric neurology group, and they may well be able to get CSF from shunts, etc., because they do a lot of shunting there. So you're absolutely right that in terms of the right dose in the right place and balancing risk and benefit in terms of going up to too high doses, I don't know about the preclinical additional work. I think you might tell me more about what you think might help with that. And I do know that there's a number of animal and other models that you can use. I think the duration story, again, a duration design where you don't say, let's compare 4 versus 6 months or something you pick out of the air versus standard of care, but actually break it down into smaller groups is something that our methodology group are doing quite a lot of. So, for example, we've got with Mike Sharland's group, an antibiotics trial in pneumonia in children, where you look at, instead of choosing 3 days versus 7 or whatever, you go 2, 3, 4, 5, 6, 7 days, and then you have a duration response curve to that. And I think that those kind of methods are going to be important for actually picking what the right duration is, rather than guess, we might be able to reduce it by this much. And those kind of methods, I think, are appropriate for those phase 3 trials. Obviously, for earlier phase trials, one has to try and think of surrogates, which everyone's still looking for, to actually inform earlier stage trials. I hope I've answered that a little bit.
Thank you. Yeah, very much. And then the last 2 questions. One just asks, why do you think there might have been a higher refusal for lumbar puncture in some countries? Zambia and Zimbabwe are mentioned in the question. Is there something cultural?
Yeah.
Or?
Well, I think, it's very interesting, because I do remember in the early days of doing PK studies for HIV in children, that it really is important to engage with communities and actually talk about what this is about, and if they see benefits of children being in studies, etc., which has often been the case, then actually that can make quite a difference. I know that, for example, colleagues in Malawi, when they were doing lumbar punctures for any reason, if if a mother came in and really refused, there would be almost a little batch of families said, no, no, we don't want to. So hence the need perhaps for some really appropriate film-based, animation-based, whatever materials for families. And we're working with the teams on the ground and the families. So this doesn't just affect TB meningitis, of course. It's much wider than that. You need to be able to diagnose meningitis and cerebral malaria, or have the child got both. So I think it's and it was different in different sites, and it was quite noticeable that in Vietnam and Uganda, which are more established research sites, rather than the general pediatric ward, they had, if you like, worked on this and already overcome that problem.
Thank you. And final questions from Willem Hannekom. So again, in the TBM study, would you be able to do some genotyping association studies with the data that you have?
Yes, I think there's possibilities of that. With Robin Basuroy and also the Vietnam group are hoping to collect further samples to do some biomarker work. We obviously recognize that these children and these samples within a complex trial like this are extremely valuable. And so, you know, have been thinking about sub-studies and what needs to be collected. On the other hand, you know, from the trials unit point of view, getting the trial off the ground and recruiting, it's an important balance that is our #1 priority at the moment. But Willem, please do contact us. And I think that those kind of things are important, and I think that we need to have more conversations with the adult groups who are running trials in TBM for adults.
Thank you. Well, Di, thank you so much again for a really fantastic presentation. I really, really enjoyed well, you did exactly what we asked you to do, to describe how trials have happened, and some that are obviously much harder to do as they are being set up through COVID. The implementation work that you're describing is really so elegant, and it's fantastic to see how fast things move in terms of WHO responding and saying, okay, let's introduce this. It would be also great to get you back at some point to hear how the other trials are going and what the uptake is once SHINE is really being implemented globally. So I know everybody, as I say, the comments you've had on the chat are fantastic. So thank you so much indeed for your time. Thank you everybody for attending and for your questions, because it always makes everything so great to see you. All the best.
Well,
thank you.
Thank you for inviting me. Pleasure.
Thank you.
Thank you.
Bye, everyone. Bye.
Bye.